I'm Megan Allen. I'm Scientific Director of Damon Runyon, and welcome today. And I will pass it over to the Damon Runyon staff, and then we can sort of ask Dr. Bird, Dr. Groshin, and Dr. Jan to introduce themselves as well. So Claire?
Hi, I'm Claire Cahill. I'm Chief Administrative Officer at Damon Runyon. Welcome, everyone. Thanks for joining.
Shannon?
Hi, I'm Shannon Donovan, Award Programs Manager at Damon Runyon.
And Sarah?
Hi, I'm Sarah Jordy. I'm the Award Programs Coordinator here at Damon Runyon.
Great, thank you. And Dr. Bird?
Hi, Kristen Bird. I'm a professor at Ohio State University and member of the Damon Runyon Innovation Selection Award Committee. And my research is focused on RAS biology as well as aging and how aging influences cancer outcomes.
Dr. Groshin?
Hi, everyone. My name is Danielle Grochan, and I'm an assistant professor at the Scripps Research Institute in La Jolla, California. And I'm in the Department of Integrative Structural and Computational Biology. So I'm the representative fundamental or basic researcher of the group. And my lab is obsessed with studying mitochondria and how they change shape under different physiological conditions or stress.
Thank you. And Dr. Jan?
Hey everyone, my name is Max Jan. I'm an assistant professor at Mass General Hospital and Harvard Medical School, the Department of Pathology and the Cancer Center, my main affiliations. And our work is mostly focused on developing mammalian synthetic biology and cellular immunotherapies.
So once again, thank you for attending the Damon Runyon-Rachleff Innovation Award info session. We are recording this session and we will place the recording on our foundation's YouTube channel. And so after the session, once we upload it, we will also send out an email with a link to the recording so that you can review it and also you can share with others at your institution. And so first, a little bit about Damon Runyon. So we're always telling people about the foundation, and we're finding that inside the cancer researcher community, we're quite well known. However, sometimes outside of that niche community, the reputation of the foundation is less known. So at Damon Runyon Cancer Research Foundation, our mission has always been to fund high-risk, high-reward cancer research. And so we identify and enable young scientists who are brilliant and bold enough to go where others have not. And we've been doing this since 1946. And so how our foundation received its name and how did we get started? I think it's most easily sort of described as it came about because of a very strong friendship between Walter Winchell and Damon Runyon. So as you may know, Damon Runyon is the famous playwright who wrote Guys and Dolls, for example. And Walter Winchell was his very dear friend who is sort of given the moniker of the inventor of gossip journalism. And so when Damon Runyon died from throat cancer, Walter Winchell was deeply affected. And he went on to his radio program and said to America, send us your dimes and your nickels, and we're going to fund the bravest, boldest, most innovative thinkers in cancer research. And so the start to our foundation all began with just collecting dimes and nickels from donors across the United States. And so today, we are a public charity that raises donations from across the nation, as well as from a few significant partners that I'll talk about later in the slideshow. And thinking about Damon Runyon, the Cancer Research Foundation, our alums, as well as the legacy. So since 1946, we have funded around 4,000 scientists with an investment of nearly $450 million. This year, we commit to give close to $20 million across 8 award programs. And so looking at some of our former awardees, we're very proud to state that 13 are Nobel Prize winners. 41 are National Academy of Medicine members, 97 are National Academy of Sciences members, 7 have been awarded the National Medal of Science, and 15 have received the Lasker Award. So if you join our sort of community, you'll be connected to a network of very prominent scientists. And the impact of the foundation and what we have funded in the past is multifaceted and bridges several research domains. So this is a very boring slide. It's very wordy, but it's just meant to sort of hit home that we fund across the spectrum. So in the past, we've funded therapeutic innovation, which you can see here, there's several drugs that have come to market that our scientists have played a critical role in presenting to patients. We also play a role in the technology innovation. So for example, we have our scientists were key very early on in CRISPR and genome sequencing, for example. And then also former awardees have gone on to co-found or start several biotech companies. And so, as I mentioned before, we're a public charity, so we are funded by individual donors. We also are funded by scientific companies and elite nonprofits that support our programs. So if you look here to the left, here are a few of our partners. So HHMI, the Mark Foundation for Cancer Research, as well as Merck. And so 100% of all of our donations are used to fund cancer research. And these biotech and pharma companies such as Merck understand how crucial your work is to the development of next generation therapies. Excuse me. And also elite nonprofits such as HHMI and the Mark Foundation also recognize the importance of co-funding. And so our award programs support the entire spectrum of cancer research. If you look here, this is just a brief schematic where we talk about the award programs that would fund basic research, translational research, and clinical trials. So on the basic research side, we have several of our fellowships for postdoctoral trainees. And then we also have the Damon Runyon-Rachleff Innovation Award for early career investigators that we'll talk about more in depth today. This also spans basic and translational research. For investigator-initiated clinical trials, we have 2 awards for physician scientists. The one is for physician scientists who are early career assistant professors, and that is the Clinical Investigator Award. And then for sort of that postdoc fellow, last year fellow instructor level, we have the Physician Scientist Training Award. And then most recently, we introduced the Scholars Program for Advancing Research and Knowledge. This is a very important program that is for post-baccalaureate scholars. So we host the call for post-baccalaureate scholars, and then we place them for 1 year to train with a current or former Damon Runyon scientist. And we have 8 partners across the United States for this award. Now, if you look at it on the flip side, our award programs support researchers at nearly every transition point in an academic career. So as I was briefly speaking about for recent graduates, we have the post-baccalaureate program, SPARK. For postdocs, we have 3 fellowship awards. For physician scientists, we have the Physician Scientist Training Award, Clinical Investigator Award, and then of course, for assistant professors, the Innovation Award, as well as the Clinical Investigator Award. And typically we fund early career investigators with pioneering new ideas. And so this is a very early photo of Danielle when she was first awarded. And our investigators, they come from institutions across the United States. So if you look here in dark blue, that's where we are currently funding. If you are from one of these states that is not in dark blue, we 100% encourage your application, and we look forward to funding investigators all across our country. So the Damon Runyon-Rachleff Innovation Award. What I tried to do here is not present to you everything from our eligibility section of our website, but to pull out some of the highlights to sort of help you figure out what to write and how to pick a project. And later you'll hear from Dr. Bird, Dr. Grosjean, and Dr. Jan more about the projects and sort of the scientist side of applying. So the program supports projects that have the potential to lead advances that will significantly impact the prevention, diagnosis, and treatment or basic understanding of cancer. So while the foundation adopts a very broad definition of what is cancer research, for these awards, you will need to write the proposal in a way that it is relevant to cancer research. Now, the overall goal of the award is to provide extraordinary early career investigators or researchers who have innovative ideas but lack that sufficient preliminary data to obtain traditional funding and or that major, your first R grant from the NIH. This award is really not designed to fund incremental advances, and then we suggest that you not write it. So we're not the NIH. We don't want you to write us an NIH grant. We're looking for those really bold, innovative ideas. And so, of course, this is an assistant professor award, and your position must be tenure track or equivalent. So pretty much as an investigator, you must be independent. We do allow senior clinical fellows who are taking a year to be independent to apply, and we also allow distinguished fellows to apply as well. So stage 1 funding provides $400,000 across 2 years. You can then competitively apply for stage 2 funding, which provides another 2 years of funding. And these funds are made to be intentionally broad to help you start your labs. So the funds may cover salaries for you or support staff, special equipment, supplies, and other miscellaneous items that are required to conduct the proposed research. So if you are interested in applying, you can go to Proposal Central and search the open applications under Damon Runyon, and you'll find the Damon Runyon-Rachleff Innovation Award. The deadline is by 4 p.m. Eastern Daylight Time on July 8th, 2024. Now, if you wanted to view more of the eligibility requirements and the terms of the award, you can go to our website and click on For Scientists, find the tile for the Damon Runyon-Rachleff Innovation Award, and you can click on Overview. That specifically lines out all of the eligibility and the terms of the award as well as funding. So this is an open call. There are no, you do not need to have, it's not a limited submission, so you do not need to be nominated by your institution. Now you can review the application components and templates. You can once again go to the website, go to For Scientists, and then you can click on Application Guidelines. So there you'll find the templates and the different components that are required for the application. And then those are also going to be in the Proposal Central. To view the currently funded projects, you can go to the website and click on Current Projects. And so that sort of gives you an idea of all the various research that we fund under this mechanism. Now, as the scientific director of Damon Runyon, I never miss an opportunity to also plug for other awards. And so as early assistant professors, you may have trainees coming into your labs. And so just keep in mind and spread the word to your trainees that we have the Spark Award for post-baccalaureate researchers. This is funding for 1 full year for students who have graduated with a bachelor's within the last 36 months. We also have 3 fellowship awards for postdocs. So we have our traditional fellowship award here, and this is $300,000 spread over 4 years. We have the Quantitative Biology Fellowship Award, and this is for computational scientists who want to ask a biological question, and it requires 2 mentors, 1 wet lab and 1 dry lab. This is a $240,000 award that's spread across 3 years. And then lastly, we have the Damon Runyon St. Jude Pediatric Cancer Fellowship Award. And so that award is for pediatric cancer researchers, and that is a $300,000 award that's spread across 4 years. And it was developed with the generous support of St. Jude. And now lastly, our awardees join a scientifically rich community that offers many opportunities. So hopefully our grants and fellowships and other awards provide the awardees with the scientific freedom to ask the questions that are most interesting to you and that may have the biggest benefit to the community as well as patients in the future. There's built-in mentorship as well. We offer annual meetings that are made for networking and mentorship. You also receive feedback from the committee when you come in to report, as well as some career advice. Many of our awardees find each other at these meetings, and it begins a really nice set of collaborations that sort of span decades. Also, we encourage the sharing of ideas and data to help each other so that you're not doubling efforts. And then lastly, we provide several opportunities just for you to network with top researchers in the field. And so, as always, you met a few of us today. So you've met Claire, you met myself, you met Shannon, and you met Sarah. We are also led by a fantastic president and CEO, Dr. Young Lee, who is also a former awardee. And so our team is pretty lean. If you had any questions, you can always email awards@damonrunyon.org, or you can email any one of us individually, and we'll make sure to get back to you. Okay. And so with that, I'll stop and I'll sort of hand it over to Dr. Bird. And hopefully that was a nice sort of introduction that will help with your slides as well.
Great, thanks so much, Megan. And let me share my slides here. Okay, can everyone see them? Please.
Yes.
Okay. All right, fantastic. So I'm here to provide a little bit of background from the side of the award committee. And so just so you know, this is kind of the application timeline. I think a lot of even awardees aren't aware of what happens behind the scenes, but you'll submit your project and then the Damon Runyon staff will review it for eligibility criteria. And then after that, the award committee will see your abstracts and kind of pick out the ones that they are most interested in looking at. So if you look at the current list of the award committee members, you may have an idea of who might pick out your abstract. But at least usually 2 to 3 reviewers are assigned then by the Damon Runyon staff. And the top applications, just like at an NIH study section, are then discussed among all of the reviewers. During this discussion, there are about usually like 8 applicants, 8 to 10 that are invited for a 2-slide concept pitch in the fall, where you can talk about your concept and how it would impact cancer research. And then there's a discussion by the committee, and the Damon Runyon selects and notifies the awardees based on that discussion. So let's see here, if we can get this, there we go. So what are we reviewing and what criteria are we looking for? The 3 main areas that we're scoring an application are on the applicant's capacity to conduct bold, exceptionally creative research, the novelty and creativity of the proposal, and the potential impact of the proposed research. And as mentioned by Megan, we're really looking for high risk, high reward proposals. So going more in depth into these 3 areas, talking about your capacity to conduct bold, exceptionally creative research. Places that we're looking for this is not necessarily that you've published Nature, Cell, and Science papers, but that you're able to articulate the impact of the papers you have published on the field and beyond. And it's important that you be able to explain how your published work took a daring approach, challenged current dogma, or produced some kind of technology that moved the field forward. It's also good, we often look for in your references for some anecdotes about how you have contributed individually to these bold and exceptionally creative research ideas, so that we know that it's really coming from you and not a prior mentor or things like that. And then the other thing that we really want to see in the applicant's background is some rationale as to why they're the only person that could potentially tackle this proposed line of research. What makes you unique? Do you have some multidisciplinary background? Are you bringing some technology or technological expertise into the cancer field? Are you approaching the problem in a different way? We just want to see how you are better poised for success than others in the field. So be sure to articulate that. As far as the novelty and creativity of the proposed research, I think this is where most people kind of stumble and aren't quite sure what's innovative or what's considered innovative. And so we want to know why your approach is outside of the box or high risk, high reward. And so we kind of see 2 different categories of this. One would be a technological kind of outside of the box, high risk, high reward application, and the other would be more conceptual. And so when you think about technological, I think you would want to say, you know, there is this gap in the field that we have been unable to address without a creative or new solution. But I have a solution, you know, you're proposing this new solution, and I'm the best one to tackle this problem because, and then explain why typical funding agencies might not be interested in that particular approach. For a conceptually novel application, you might be stating that you're thinking about a problem from a completely different point of view that has yet to be addressed by the field. And explain, maybe your view is partially supported by the literature, or maybe you have one or two pieces of preliminary data, but at this point it would be considered way too risky or way outside the box for a traditional funding agency. And so I think if your project fits in one or two or both of those categories, then it's probably appropriate as far as its novelty and innovation. The last category is its potential impacts of the proposed research in the cancer field. And so we're hoping to see something that will potentially be applicable to more than one cancer type. So it doesn't necessarily have to be that your proposal is on multiple cancer types, but that it's a concept or a thread that might run between different cancer types. That it's going to result in, if it's successful, it's going to result in a major conceptual or technological shift in the field and enable the advancement of cancer research on multiple fronts. The other thing is, you know, we're always asking ourselves, if this person doesn't do it, will it get done? And a lot of times we're saying, yeah, if this person doesn't get this money, it's never going to get done. And so if you have a project like that, I think the committee gets really excited about those types of things. So that's where I'm going to end there. And because I definitely want to give the chance of the innovators to give a little bit of background on what they've been doing, and then also for you to ask questions. Should I throw it back to you, Megan?
Yeah, sure. So I guess in the program we'll ask Danielle to start, please. Thank you.
All right. Hello, everyone. I don't have any slides, but I can give just a basic overview of the project that I proposed. I applied in 2020, and so I'm coming up in my last year of Damon Runyon funding, and I have to say this award was very special to my lab because it was the first grant award that my lab received. So it'll always be near and dear to my heart. But the scientific aspect of my proposal was really focused on mitochondria and how in cancer they tend to exhibit hyperfragmentation. So what does this mean? This means that under normal homeostasis, cellular homeostasis, mitochondria are quite dynamic. They constantly undergo cycles of fusion or joining together, or fission or division. And this is necessary to maintain a healthy population of mitochondria in the cell and as a way to achieve mitochondria quality control. But under certain conditions, and this is quite often seen in different cancer cell types, you have hyperactivation of that fission pathway. And this leads to mitochondrial dysfunction. And just taking a step back, this is sort of one of the new hallmarks that's thought of mitochondria and their connection to cancer. Obviously, there's the well-established Warburg effect, which is almost, I think, nearly 100 years it's been known about the Warburg effect for cancer cells, where they have decreased reliance on mitochondria energy and increased reliance on glycolysis as a way to sort of bypass that regulation. But mitochondrial fragmentation is recently emerging as another sort of hallmark that maybe we should pay attention to as a strategy to actually target and prevent tumorigenesis. And so there are several players that are known to be involved in the mitochondrial division process. One of the key players is a protein called DRP1. This is a dynamin-related protein. And it's thought to be the main protein that's involved in the force generation to actually pinch off and divide the membranes, leading to division. Now, what's really interesting is that DRP1 can be regulated in many ways through post-translational modifications. And there's a specific site on DRP1 that is phosphorylated in different cancers like ovarian cancer. And this hyperactivates DRP1 and leads to that fragmentation observed in cancer. And if you inhibit that phosphorylation and that hyperactivation, you can actually prevent cell metastasis. So this has really opened up this exciting idea that maybe we could therapeutically target DRP1 to prevent this uncontrolled mitochondrial division that's exhibited during cancer. But the challenge is that despite several efforts, we still don't really have a great inhibitor of DRP1. And part of the additional challenge is that we still don't really understand beyond post-translational control how DRP1 is regulated. How does it know where to go on the mitochondrial membrane? How does it know when to exhibit its constriction function? And so my lab in this proposal really wanted to tackle this, and we hypothesized that there was a hidden level of structural regulation that was mediating DRP1's action. And so we wanted to uncover this by harnessing some of the recent advancements in the structural biology field that enable us to actually dive inside of cells and solve high resolution structures of proteins in their native cellular environment. So maybe some of you have heard of cryo-electron tomography, cellular cryo-electron tomography. This is the approach that we want to harness to visualize for the first time constricting mitochondria and DRP1 in action. And so my proposal basically outlined my plan to harness this technology and to provide the first glimpse of this machinery, both that maintains cellular homeostasis, but also in cancer. And our long-term vision is to be able to leverage the structural insights that we gain from looking at these snapshots and eventually look for new interactions, structural interactions or assemblies that we might be able to therapeutically target with structure-based drug design. So that's all the science. Maybe I'll hand it over to Max for his bit.
Okay, thanks so much. Hey everyone. So I applied in 2022, and that was in my first year leading a lab. I had left a postdoc in 2021, and that postdoc was really focused on trying to, it was dual mentored, and it was focused on trying to create a new set of links between the field of targeted protein degradation drug development as well as cell therapy. So chemogenetic control of cell therapies and ways to have more tunable, precise approaches to treat cancer with engineered cells. And then this was very early days, and was really grateful to have a mechanism that was sort of well aligned with where we were. So we came to the Innovator application process essentially with a new technology. So having developed a new technology for transgene encoded targeted protein degradation, so you could engineer only a subset of cells, anti-cancer T cells, to our goal is to have them be able to redirect E3 ubiquitin ligases to degrade proteins, sets of proteins with multi-specificity that limit the anti-tumor activity of those engineered cells, CAR T cells, for example. So, kind of a platform that could be applied to different cell types and also in different cancer settings. And aims 1 and 2 were really 2 different applications. One to help with tumor antigen recognition, and then aim 2 was to engineer cells to be resistant to suppressive signals in tumor microenvironments. I could go on and on about how wonderful it's been to be a part of this community, and it really has been incredibly impactful. It's also a really interesting and pretty unique application process. So happy to talk at length about those things, but definitely want to hand it back to Megan to see where we should go next. So thanks.
Yeah, great. Thank you so much. And so Max and Danielle, so Max, you mentioned that you applied as a first year assistant professor. And then Danielle, you also applied very early into your assistant professorship as well. Is that correct?
Actually, I applied as a fellow. So right after earning my PhD, I started my independent career as a fellow. And so I'm thankful that this award is there's not that many independent funding awards for fellows, but this was one of them. And so I applied within my first year of the fellows position, just right after my graduate training.
Great, great. And then, so we get the question a lot, should an assistant professor wait or should they apply when they have this idea? And I think you both are great examples that we do fund that first, second year assistant professor. You don't have to wait until your third, fourth, or fifth, for example. And then very quickly, if I could just ask both of you to sort of talk about, so we have a stage 1 and we have a stage 2, and we have that written in our eligibility. So how did you personally write your proposal? Did you only focus on a stage 1, or did you sort of touch upon what you would do for stage 2, for example? And then also, yeah.
Yeah. I initially just wrote it for that stage 1. But having said that, I definitely emphasize the big picture, like, goal that we had, which would carry me through stage 2. But initially it was just focused on the bold idea, the application of new technology for that first stage funding.
Yep. And Max, how did you handle that in your application?
Same here, like really kind of hand to mouth and really just focused on the first stage, and my second stage application is coming up in the next few months, so we'll see about that.
And then Kristen, if we could hear your sort of opinion from the committee side, because we don't provide any strict structure, we allow the applicants to choose how they prefer to write it. And then so from your side, how do you typically sort of want or prefer to see that in a proposal?
I think you can structure it however you like. It seems that, it's very proposal specific and what you're specifically looking at. However, I'd say that most of them come in as kind of a very ambitious 2-year proposal. Pretty broad and ambitious, kind of highlighting those big picture questions. And the thought there being that, whatever doesn't get done in those first 2 years will be in the next stage. So I think that's kind of how the committee usually sees these as, a presentation of an initial idea.
Great, great. Well, thank you very much. So what I will do is I will open up the Q&A portion of the session. We received a few questions beforehand, so I can start with those. And for the participants, if you have any questions, you can either write them in the chat or raise your hand, and you can, once I call on you, you can unmute and ask the question to the panelists directly. Great. So this one I think is probably appropriate for all three. But do you have any advice for applicants who have an innovative or bold idea, but they have several of these ideas? How would you sort of pick one? And so we get asked this quite a few times each cycle. Maybe we could start with Dr. Bird.
I would say to go around to some of the people at your institution and kind of pitch the ideas. Try your elevator pitch for each one and see which one makes the most number of people excited. The committee is pretty diverse in what they do and what they're knowledgeable about. So you want to make sure you're not just talking to people in your area, but people outside. And just see, I think eventually you kind of land on something you're really passionate about, and that gets that feedback from others that they're also really excited about it. So that's what I would say.
Great, thank you. And Max? Not to put you on the spot.
I think another suggestion for those who, are in that great spot of having multiple possible application worthy projects is thinking about the timelines. And in this case, this could really be treated as two separate 2-year applications. So really thinking about what might be able to kind of show next exciting steps within that timeline might be able to narrow things down a little bit.
Perfect. And if you don't have anything, I can move on to the next question if you.
No, I think that was great advice. I would also just based on Dr. Bird's sort of what they're looking in the review process, if you can narrow it down based on what about that project is something only you can do. I think that's also a good criteria.
Yeah. Great, thank you. So one more question from those that we received prior, and then I'll get to the question in the chat. So Dr. Bird, you mentioned in the slideshow that you're not necessarily or only wanting to see those Nature Cell science publications, but really looking for scientists to discuss the impact of the publications that they have been a part of. What advice do you have for some of the brand new assistant professors who have been really busy focusing on setting up the lab and so might have a gap in that publication record? Do you have any advice for those investigators?
I think if they can find if they can, select people to write letters of recommendation who can really speak to the impact of their postdoctoral work or fellowship work, as well as kind of their personal role in that, that can be very helpful. Or people who know that, who are able to speak as collaborators or things like that, right? That know of their work. So it doesn't necessarily have to be that it has to come from your independent lab, but someone who can speak to what you personally contributed to those prior publications would be fantastic.
Yeah. Okay. Great. And then here I have another quick question. So can you sort of, even though this question is sort of thinking ahead, past that first round where you get invited to the finalist interviews, how stressful was that to prepare, and how were you able to sort of organize that sort of pitch to the committee? What could be helpful for potential applicants to hear?
I can start off if that's okay, Max. So I actually, as nerve-wracking as it was, I actually wish every grant I could get in front of the reviewers and pitch a case for that, because I think sometimes the translation to a written document versus how you can portray your excitement or the impact of your work in an oral presentation. It's sometimes, depending on your skill set, it's easier for me to do that in a presentation format. But I do have some practical tips for that. And the biggest one is just practice, practice, practice. I think I did probably 4 practice rounds. The first was with what I call my inner circle of mentors, which are people who are very familiar with my research program, very familiar with the project. They probably reviewed my application before I submitted it, so they have an idea. And that round is rough. You're just trying to get your basic, like, am I communicating things? How should my slides be organized? And then I definitely did a round with people who were, because I'm more on the fundamental biology, I did it with more sort of cancer focused labs, assistant or associate professors or full professors that have that sort of more focus on cancer, to make sure that I was really hitting that audience as well. And then the last 2 rounds, I think one of them should be with an audience that maybe is less familiar with your work and totally outside of it, because they're actually going to ask maybe the questions that are the ones that you want to get asked in a practice round, and that might come up during the real thing. And then, yeah, just having your pitch down, because there is a timed, I forget how long it is, but you want to make sure you have your cadence down. And so for me personally, it was just having lots of repetition, lots of feedback, and then settling on where I finally got to the day, and I was actually excited to tell the committee about it. Not still nervous, but more excited.
Yeah, so totally agree with all of that. I have to tell you that I found the process not unstressful, I think. And but at the same time, I just it's a really great skill to have. And you know, was I definitely will always have that experience. And you know, it's something that I think was really valuable about the whole process. The kind of biggest thing from my preparation was just learning how to simplify and how to get straight to the point, both visually and with one's words. And then what turned out to be most helpful was agree, like multiple rounds of practice, and specifically with people who have familiarity, you know, ideally with the mechanism or with similar mechanisms. And I don't know if it's true. I think probably so. I'd be happy to, you know, help, you know, review stuff this year for people in that round. Just please reach out. And then also like senior folks in the departments that I'm affiliated with who have tons of experience with things like this. And so especially because that sort of automatically made them from different areas of cancer biology research, it was pretty representative. And yeah, I think for me, those would be things.
Great, thank you. And so we also had another question here about, so, you know, I also talk about the benefits of becoming a Damon Runyon awardee. And so I think one of the most fantastic benefits is that you can come in and speak with the committee. And so perhaps, Kristen, could you tell us a little bit about when you meet with some of the innovators in that room for when you hear their science, and what kind of sort of mentorship goes on between the committee and an awardee, for example?
Yeah, so I think it's just a fantastic environment because you have this group of just really energetic scientists with all types of different ideas and technologies. It's kind of the greatest of all worlds. So I think the committee is just as excited to talk to all of you and to hear your pitches. But then, you know, there's this opportunity from the committee who's also pretty diverse in their scientific background and training to get some different ideas and perspectives that you might not hear, but also to start up collaborations or to help connect you with other people in the field that may be able to help solve a problem that you're having, or to help you with career advice and things like that. So I think that's really the fantastic benefit, is just having that group of really kind of, you know, the like-minded innovative group where people think outside the box a lot and come up with strange ideas, is just very fun and energetic. And I think it's a two-way street, both for the innovators and the mentors. We're really learning from each other.
Yeah. Great, thank you. And so we have another question in the chat here. And so this is sort of about proposing a project. So does it necessarily have to be based on previous work or published work? Or can it be a novel idea with some preliminary data and a new direction that there's not necessarily that publication record? And so for the advice what would you give the advice or how would you give advice to that type of scientist?
Yeah, so I would say that, yes, it can definitely be something very much outside of what you've been doing, but you have to show that you're the person to do it, right? I have to at least believe that you have the capability to work in that area. So, you know, that needs to be kind of sold in the application, but we're definitely seeing applications that are from people who are really transitioning fields, from people who don't study cancer. There's always a few of them in the top pile. So definitely, don't think that you have to have this long track record of cancer research in order to be successful.
Great, and so sort of a follow-up there. So, if there's no publication record and if it is sort of a new line of research, how either did the current awardees or you as a reviewer, how do you look upon an applicant putting together sort of an advisory team, a scientific advisory research team, to sort of make up for those deficiencies, if you want to call them deficiencies? Did anyone have sort of senior scientists in their application, or, how would you look upon that as a reviewer?
Did you guys, either of you, have a senior scientist?
No.
Yeah, I would say it's pretty rare that we see a senior scientist making up that gap. We have seen co-applications, so I will mention that. We've had several very successful co-applicants where you have two people who are eligible and they come together and they bring their separate expertise. So that is another option. Obviously, if you're missing part of that expertise, consider a co-applicant. It doesn't have to be somebody at your institution either. Most of our co-applicants have actually been at separate institutions. So it's very interesting, and they tend to do quite well because they're bringing two novel things together. Otherwise, if you really don't have that expertise, it can be tough, I think, to sell it as your own personal idea that you're bringing forward. If you have some preliminary data, that certainly could help.
Yeah, I can just maybe follow up with that. I didn't have submit with a co-investigator, but my sort of pathway to application was that within that first year, we had collected some preliminary data that gave me a glimpse into something that could be promising and really did form the basis of my hypothesis for the application. And I think something also that I did dedicate a significant chunk of my application to is that part which is justifying why it wouldn't be competitive for traditional NIH funding, and think beyond the fact that maybe you just don't have enough preliminary data that you need these days for an R01. I think it's beyond that. It's really like the idea is such a big jump and so bold that it would be very challenging to sort of demonstrate feasibility, even if you had a whole bunch of initial preliminary data. So I think that's something that I definitely, in lieu of maybe describing a lot of the experimental details, I spent a significant chunk of my written application on that part.
Great. We do have an eligibility question about this award. So this award is for independent researchers. And so while one of the sort of eligibility criteria is a tenured assistant professor, if you have an MD or a DO and you are seeing patients and you're in your last year of your subspecialty, and you have independence and the support of your institution, we would allow that, but you should email us to sort of check further. And then we also allow distinguished fellows, and as Danielle mentioned earlier, she applied under that eligibility. And so these distinguished fellows are sort of that the independent fellow that's offered, say, by Cold Spring Harbor or Schmitz Fellows or something like that. So there's that need for independence. We do have other awards for postdocs as well as instructors. Those are sort of on the eligibility there is depending on your degree type. So if you go to our website and go to For Scientists, all of the eligibility criteria for all of our awards are there. And then we'd be happy to sort of follow up one-on-one. If you email us, we can review the eligibility as well. Great. And so are there any other questions from the audience? All right, well, great. Well, I just want to thank our panelists, Dr. Bird, Dr. John, Dr. Grotjan. It was fantastic hearing you and seeing you. And I just remember actually both of you when you presented via Zoom, I remember each of your presentations and how exciting they were. And also how, to Kristen's point here, how exciting or excited the committee was after your presentation. So always thankful for all the help that you give to Damon Runyon. And hopefully this session was informative to the group. And if you have any other questions, please feel free to reach out to us at Damon Runyon. You can do awards@damonrunyon.org, or you can email me directly. It's megan.allen@damonrunyon.org. All right, well, thank you all very much and have a very nice rest of your day.